Hello, everyone. Thank you for joining our first Australian Heroica webinar, first of hopefully many to come. We're going to wait for some individuals still to join. So if you're joining in from the audience, please feel free to let us know where you're joining in from. So. And while we have people joining in, we're going to play some music.
Prostate cancer is an enemy, killing many men with no mercy. You can check your family history for warning signs. When you love up the world, can you raise up a sound? Or there's a want to fight, we will stop till we get it right. Men will love up the world, can you raise up a sound? Or there's a want to fight, we will stop till we get it right. Take good care of yourself, pay attention to your body. Take good care of yourself, pay attention to your prostate. There may be no symptoms of something wrong, no signs, no warning. So know your history as you're growing older. Go check your body and fight from a place of rest as you eat a healthy diet and regular exercise to keep a healthy weight. When I love the world, yeah. Can you raise up a sound? Oh, there's a water fight. Go fight till we get it right. When I love the world, can you raise up a sound? There's a water fight. Go fight till we get it right. Pay attention, yeah. Pay attention. Warning signs, warning signs.
Good morning, everyone, or good night and good afternoon. So for today's webinar, we have individuals from different time zones. So for our USA folks, I did want to say thank you for rising early to attend today's webinar. For our folks in South Africa, Kenya, thank you for this great afternoon for you attending and seeing the webinar. And again, for our folks in Australia, it is about 9 p.m. their time. So again, thank you again for staying late to see this webinar. So with today's webinar presentation, we have presenters from across the country, and we would like to specifically, within eye care, we have specifically within eye care, we would like to showcase Dr. Hayes, Dr. Miss Vanessa Hayes, and her lab and all the work that they've been doing related to prostate cancer. So I've added Dr. Hayes to the stage to just essentially just provide a kind of a welcome greeting and just to present, to showcase the presenters that will be presenting today. So please give a warm round of applause, even though we can't hear you, or welcome through the chat. Just a warm greeting to Dr. Hayes.
Thank you, Emelina. So welcome, everyone. And first of all, before I start, I really want to thank the eye care team at Mayo Clinic for welcoming us as the other heroic team to this forum. And we hope this will be one of many. So this will be our first webinar. And just to introduce us, the heroic team that is led out of Australia, we actually led by 4 of us partners. So it's ourselves. I'm Professor Vanessa Hayes, the University of Sydney in Australia. Then we have Professor Rihanna Ballman, who leads the team out of the University of Pretoria in South Africa. We have Professor Peter Ngugi, who leads the team out of the University of Nairobi in Kenya. And we have Professor Gail, who leads the team out of the University of Illinois at Chicago, obviously in the USA. So our 4 speakers today are actually from 2 of our branches of our research. So we will start off with Dr. Winston Ombuki, who will talk about his research into looking at PSA levels. And then we'll have Tabolgo Lebello, who will talk about her work on hormones. So they come from the Urology and Pathology Working Group or team. And then we will, the second half, it'll be Dr. Pamela So and myself, Vanessa Hayes, that will talk about some of the genetics work that we have been doing. So I'd like to get going so we don't waste any time. Dr. Winston Ombuki, if you'll go ahead. Thank you.
Dr. Hayes.
Thank you. I'll proceed to present on the prostate cancer presentation in Africa, lessons from a large retrospective study spanning a decade. As been told, my name is Dr. Winston Mokombuki. I'm a consultant urologist and a PhD student at the University of Nairobi under the Heroic Study Consortium. So we'll talk about the status of prostate cancer in sub-Saharan Africa. We know that there are a lot of disparities in terms of prostate cancer presentation. And in Africa, we have alarmingly very high PSAs and the late stage presentation, and the disease is actually of aggressive nature. So with the heroic this consortium, we are looking. This kind of study we looked at from 2010 to 2019. And we looked at one of the largest numbers in sub-Saharan Africa, 8,600 records, spanning from Kenya, Uganda, South Africa. And in the Heroic Consortium now, we are going to have also data from West Africa in Nigeria. This is Kenya and Uganda and then South Africa. So this study is actually essential because now it enables us to see the actual data on the ground, and this will be timely in terms of getting Africa-centered data and African-centered solutions. So we do know that the GlobalCan reports the mortality rate for prostate cancer is highest in South Africa, followed by Western Africa, Central, and Eastern Africa. But our records will show a bit of few changes. And because the GlobalCan data is actually a projection from hospital visits, and there's a lot of our projections, so may not reveal the actual data as it is. And we do know in Sub-Saharan Africa, we have limited number of studies. However, it shows that prostate cancer in Black African men commonly occurs at an earlier age and is more aggressive. So the true impact of across Africa remains poorly appreciated, and this requires country and regional validations. So to address this gap, the Health Equity Research Outcomes Improvement Consortium, HEROIC, Precision Health PCA PH Africa 1 Consortium, through South Africa Prostate Cancer Study, East Africa Prostate Cancer Study, and now we're going to involve Nigeria also. That is going to give us a more clearer perspective on the clinical impact of prostate cancer across Africa. Our aim was to look at through a decade between 2010 and 2019 of patient records, the presentation of prostate cancer across sub-Saharan Africa, with the highest being reported in South Africa and the lowest in East Africa. And our aim, really specific objectives, was to determine the average age and PSA levels at prostate cancer diagnosis, and to determine the proportion of patients who present with symptoms versus those who are asymptomatic at diagnosis. So our study design, setting and participants in South Africa, we looked at tertiary hospitals, Steve Biko and Kalafong Hospital, that is in Gauteng province in South Africa. And in Kenya, we looked at Kenyatta National Hospital, which is in the capital, Nairobi, and from a nearby region, Mount Kenya region, that's Meru Teaching and Nyeri Hospital. The distance between these two is around 250 kilometers, with Nairobi being largely an urban setup and Mount Kenya region being semi-urban. And to give a global perspective, we also matched data from Uganda, which also looked at data from 2015 to 2019. So we looked at the age, PSA, and ISOP grade group for 8,634 patients. And in South Africa, we had around 6,500 Black cases, South Africans. And in Steve Biko, Kalafong, we had 1,229. In Kenya, the majority of these 1,260 came from the capital, Kenyatta National Hospital, and then the nearby hospitals contributing to 2,263. And we also matched data from Uganda of 874 records across the same period. So in terms of the results, for South African age distribution of prostate cancer, the range in Kenya, in merging South and East Africa, is the age of prostate cancer also from the age of 22 to 102, with a mean age of 68, which is closely resembling the white in the US. We look at the SEER database, which was almost similar, but we also see that the Black Americans are presenting at an earlier age of diagnosis. But what is striking for us is that we know that prostate cancer is an age-dependent disease, but we have a majority of our patients are really from the age of 60 to 79. But we have these younger people who are under the age of 50, contributing to 2% in South Africa, in Southern Africa and East Africa. That is in Kenya, 8, less than 1, and around 1% in Uganda. Our median age in South Africa was slightly younger, mean age was 67, and almost similar in Kenya and Uganda at 72 and 71 respectively. And so in terms of the PSA, this is another point that wanted to highlight, is that majority of our patients have actually high-risk disease, with high risk being PSA of more than 20, but we even have PSA going to up to 1,000, above 1,000 in South Africa, contributing 8%, in Kenya around 7%, and in Uganda around 14%. So this is different from the USA data, where we're seeing the high-risk disease contributing to around 17%, and in Southeast Africa, contributing around almost over 60%. So I chose the colors of orange to show that orange is bad and the blue is good. So this is what we'll maintain throughout this presentation. So we also looked at, we know that age is a risk factor for prostate cancer, and we really wanted to see whether age is a confounding, if there's any risk association between age and grade group, and we realized in terms of the age is not a confounding factor, and whether the age is associated with the high PSAs. This was more pronounced in South Africa, where older men presented with higher PSAs, although even men under 60 will have higher PSAs, but this was more pronounced in South Africa. But in Kenya and Uganda, it was almost similar, although Kenya and Ugandan men were diagnosed at an older age. So they're still maintaining that the orange is bad and the blue is good. We have in East Africa and South Africa, and really merging the two, we have more pronounced higher risk group of the aggressive disease, ISOP grade 4 and 5. It's really more pronounced as compared to white US and the Black US, where we have more of the least differentiated grade group 1, 2, and 3. So this really limits the treatment options for our patients when they present with advanced disease. And now we also have these PSA levels, where the orange is really the bad one. We can see this more pronounced in Southeast Africa when we match the three countries compared to white US and black US. So it's really showing that Sub-Saharan Africa men present with very high PSA levels. So in terms of the pathological features and trying to see whether the BPH, how they present. So South African cohort showed slightly high prevalence of BPH, around 41%, and prostatitis about 33%. And we know some studies are ongoing about the role of inflammation. And while regional match data for prostatitis appears lacking, Ugandans reported a prevalence of around 22%, and with a global study from 2019, with a reported BPH prevalence of 63% for South and 68% for East African men, which is almost similar. And also trying to check on whether what really brings these men to the hospital. Majority of these men, 62% from Mount Kenya region, have at least symptoms at presentation, meaning the majority will come when they have symptoms, and 38% are asymptomatic, which points towards probably low screening rates when they have to really have symptoms to come to the hospital. And our lower prevalence in our study may point towards a difference in cohort, presence of prostate cancer, and the absence of reporting. So in terms of the conclusion, we are looking at it's time for us to shift the paradigm from late stage detection to early intervention, and the overall goal being to reduce mortality across sub-Saharan Africa. And our study findings emphasize the limitations of prostate cancer screening criteria. We know in most of our data we use the American and the European guidelines. And it's really this large study, one of its kind, shows us that the burden may be underestimated, particularly in East Africa, and it's time to really have Africa-centered screening guidelines, which will necessitate for regional specific adjustments. And our aim is really to improve the overall health and well-being of men across sub-Saharan Africa, and it's time for us to give prostate cancer the attention it deserves. Thank you.
Thank you, Dr. Mbuki, for that great presentation. I'm looking through the chat to see if there are any questions, and I do see some questions within our chat. So our first question we see here is if you experience any challenges in data collection.
Yes, we do. Challenges in data collection?
Yes.
Yes. So thank you. So in terms of the data collection, this was a retrospective study, and we know that even the ISUP grading has changed over time from the 2000s, and so some of the challenges we faced was incomplete data entry, and we do know that most of our hospital records are manual. And I think moving to the digital will really help in storing our data. But so some of incomplete records, and we really try to only include the complete data.
Okay, because that was a follow-up question. If so, how have you addressed those challenges? Our next question that we had was data record, so patient records, manually obtained?
Yes, patient records was manually obtained, and that is we went actually through the records through the time we got through the hospital records. And in terms of how to mitigate or improve on our recording or reporting, we now have multidisciplinary teams where, for example, in the urological community, we get together with the pathologist and try to standardize our reporting so that it is more or less the same across all hospitals.
Great. Thank you, Dr. Mbuki. For that great presentation. We will move on to our next presenter. So, our next presenter is Toboko Labello. Did you want to try to see if you could share your slides? If not, I have them open too.
Okay. Can you see them now?
Yes, I can see them. It's not full screen.
Okay, can you kindly share on my behalf
Mm-hmm.
if it doesn't show full
Do
screen?
that.
Right.
Give me 1 moment. Okay.
Thank you so much. Good afternoon, everyone, and good evening from everyone from all over the world. As Prof. Hayes has introduced, my name is Tawa Palivelo from the University of Pretoria in South Africa. So I will be taking over from where Dr. Umbuki has left off, but looking specifically at androgen levels in Black South African men, specifically testosterone. So I'm just going to give you a brief background in the next slide. So testosterone is the main androgen in males, specifically responsible for male characteristics. And also testosterone levels physiologically decline with age. Therefore, testosterone deficiency is quite common in men of older age. So the link between prostate cancer and testosterone is quite controversial, as all the studies do indicate that there is a link between prostate cancer and total testosterone. However, a recent study do dispute that. High testosterone levels, rather, at a younger age are associated with prostate cancer at older age. This means that men in their peak ages, if they have very high testosterone levels, this put them at a higher risk of prostate cancer when they age. But we now know that the total testosterone is not associated with prostate cancer. However, lower free testosterone is associated with prostate cancer risk. Interestingly, we see that there is racial disparity in the testosterone levels, with the men of African ancestry having higher testosterone level compared to men of European ancestry. So I'm just going to give you a brief background on PSA in the next slide. Can I have the next slide? Thank you. So PSA, or prostate-specific antigen, is the oldest biomarker used for prostate cancer diagnosis and management. And then also we know that PSA increases with aggressive prostate cancer. With it is quite higher in older men, because men, as they get older, they are more prone to prostatic disease. And we know that PSA is not specific to just prostate cancer. So high PSA has been inversely associated with testosterone, meaning that when men that are at high risk of prostate cancer, they might have high PSA levels, and they will have lower free testosterone levels, which is indicative of a prostate cancer risk. So just like testosterone, the PSA levels are different between men of different ancestries, with African men having much higher PSA levels compared to men of other ancestries. So before I go into the results, in the next slide, I will introduce you to the Southern African Prostate Cancer Study. So the Southern African Prostate Cancer Study, or SAPCS, was established in 2008 in South Africa by our study leaders, including Prof. Hayes, with the main aim of identifying the genetic and non-genetic factors which contribute to aggressive disease presentation across Southern Africa. So this is also the largest African prostate cancer resource, with the prostate cancer resources. What we do is to collect different data, including clinical data, demographic, epidemiological, and genetic data. So now I'm just going to give you a snapshot, show you pictures of what the study collection and going into a study site looks like. Okay, so if we look at the top left, you'll see that we have older men. So in our communities, most of the communities that we collect our data from and recruit men from are quite rural. So we have to first introduce ourselves to the tribal leaders. So most of them being men. we introduce ourselves to the tribal leaders, work together with our community health workers. We introduce ourselves, and they are the ones who connect us with the community. In the middle, we have our urologists. So we have doctors that will be doing the biopsies. And if we look in the bottom center, we see that there is a community health worker who is speaking to an elderly man. And these are the people that help us to be able to collect the data or fill the questionnaires, communicating with the men in their preferred language. On our top right, we have our project manager, who is very important in the running of our different projects in Southern Africa. And that is some of the samples that we might store. In the bottom right, that is the people that includes the study leader, which is Professor Borman. And then in the middle, we have a nurse that is also who plays a very important role in collecting some of our clinical data as well. And then the gentleman there is one of the tribal leaders, who connects us to the community so that we can work together in having prostate cancer awareness and also helping the community. So now I will go into the results. So in this table, we are looking at the different testosterone levels of healthy men. These are men without prostate cancer. So we are looking at total testosterone, free testosterone, and bioavailable testosterone. You will notice that on our left-hand side, we have the younger men, which are men less than 60 years of age. And on our right-hand side, we have men that are a bit older, which are men more than 60 years of age. You will notice that there is a difference between the white US men and the black US men, with the black US men having higher testosterone levels compared to the white US men. However, black South African men have much higher testosterone levels, both at younger age and older age. Interestingly, we know that when the men age, their testosterone level go down. But interestingly, we see the decline is more shallower in men from South Africa. From men from the US, black and white, we see that there is quite a steep decline in the testosterone level when we look at the young and older age. However, we see the much shallower decline, which is more prominent in bioavailable or more significant in bioavailable testosterone at the bottom. If you look at the bottom right, the fold increase is 1.14 compared to the 1.6 and 1 of the Black American and also the white American men. So now I will just show you the different testosterone levels in our South African cohort of 250 men. Okay, so this just shows us the different, it's a lowest curve of different testosterone levels, from total testosterone being on our far left, middle being bioavailable testosterone, and far right being free testosterone. What we see is that there's a steady trend and a difference between both the men with prostate cancer and men without prostate cancer. The men with prostate cancer being the ones in a blue line, and ones without prostate cancer is the red line. An interesting thing that we see here is that at an elderly age, specifically at 70 years of age, we see a sharp decline in men with prostate cancer. Their testosterone level drops quite sharply. If we compare from the younger age, we see that there is quite a difference between the men with prostate cancer and men without prostate cancer. However, when we get to 70 years of age, there is a distinct difference between the men with prostate cancer and without prostate cancer, because the testosterone level drops sharply with the men with prostate cancer. And we see this in the total bioavailable and free testosterone levels. Okay, I will go over to the next slide and then just show you the difference when we divide these men in different prostate cancer risk groups. We have in the middle the high risk prostate cancer group. So these are the men with a higher Gleason score of 4 or above, or with if the Gleason score 7, it's 4+3. And also with a higher PSA. So we see that there is a significantly lower testosterone level for the men with high-risk prostate cancer compared to the men with low-risk prostate cancer and no prostate cancer. Now, interestingly, we see that the men at 70 years of age, there is a significantly lower level of testosterone for the men, especially for the men with high-risk prostate cancer. So this just explained that sharp decline that we've seen in the previous slide. So now I'm just going to quickly look at our PSA levels and see how that can actually explain what we are seeing. So here we see that the men, when divided into different risk groups of prostate cancer, as explained, men with prostate cancer will have higher PSA levels. So here we see that the men with high risk prostate cancer have higher PSA levels. However, most of them have a mean PSA of over, or a median PSA of over 20 nanogram per mole. This is quite high for our PSA. And then when we look at the specific men from South Africa from different ethno-linguistic groups, we do not see a difference between the different ethno-linguistic groups, which actually highlights that all of the men from South Africa just have very high prostate PSA levels, and those with prostate cancer have significantly higher PSA levels. So now I'll just give you a summary of what I've just presented on in this particular study. So in summary, we've seen that Black South African men present with much higher testosterone level globally. With these Black South African men, especially men of that have prostate cancer, they have about 1.4-fold higher total testosterone or testosterone overall. And also we've seen the age-associated decline in testosterone, especially in men with prostate cancer or the prostate cancer cases, which is more significant in men of 70 years of age. This difference is actually very important in addressing the androgen deprivation therapy protocol for the men of African ancestry, specifically men in South Africa. And also the very high PSA levels in the Black South African men is very, very worrying, especially in men with high-risk prostate cancer. However, something that is very interesting is that in this particular cohort, there was no association between testosterone and PSA, which basically just says that the testosterone levels that we are seeing are not the drivers of the PSA. However, we know the association with a high-risk disease. This is very important in addressing the prostate cancer disparity that we see in men of African ancestry, specifically in men from Southern Africa. Thank you.
Thank you, Ernst. Welcome for that great presentation. Super insightful. I really like looking at the graphs and science behind it. So, super great presentation. We do have some questions for you within our chat. So, first question was, did you measure estrogen levels?
No, we did not measure the estrogen levels. We looked specifically just at the testosterone level, total testosterone, and then we also looked at albumin and sex hormone binding globulin so that we can be able to calculate the free and bioavailable testosterones.
Our next question that we have was there a significant difference between the levels of testosterone for Black USA versus Black South Africa?
Was there a significant difference between
Blacks who reside in the USA versus Blacks who reside in South Africa?
Yes, there was a significant difference. That's why this was very interesting, because we've just known that men of African ancestry have very high testosterone levels. However, now when we looked at men from South Africa compared to Black men from the US, we saw that big difference in the testosterone levels.
All right, our next question was, how did you navigate discussions with the tribal leaders to get their support for the study? Did you experience any issues initially? And if so, how did you manage these?
Initially, thank you for that question. So initially, what we had to go and just study the community, or from the hospitals that we work with, we have our nurses that actually reside in those different communities. So we work together with the nurses. They are the ones that actually go before us and introduce us to the tribal leaders, so that it does not seem that there is an outsider that is coming in and seeming like a threat. So now we have the health workers that are from those specific communities that go and introduce us. And that is what makes the community leaders to be more open to actually work with us, because we did not go before the people that they live with. We sent out the community leaders which work and live in that community to go and express our research interests. And then once the door was open, then the community leaders were able to actually welcome us and let us address the communities. So most of the people that work or take the questionnaires, they are actually the people that reside in those specific communities, and they are able to actually communicate in the same language.
Got it. That's great. Thank you. We have a couple more questions, but I want to be cognizant of time. So, um, the next question I'll ask you. So, piggybacking off of the question that said, was there a significant difference between the levels of testosterone for Black USA versus those from Black South African? The follow up question was, what could that difference mean in ML? Would you happen to know that information? No.
What does that difference mean? So we do not know exactly what causes that difference, but we know that men of African ancestry generally have higher testosterone levels. However, men of Southern African ancestry. So this is because we have different ancestral groups. We have men of Western African, men of Eastern African, Central African. So this could be different. It can be multifactorial. So it could be either lifestyle, either environmental, genetic, and also other socioeconomic factors that contribute to these. So that is why we are seeing these different testosterone levels from men, even though they are of African ancestry. We do see that they have high testosterone. However, when we compare between men that are geographically in different areas of Africa, then we will still also see the difference. And that is why we see this difference, or that is what can explain the difference that we see from men of African ancestry that reside in the US and those that reside in South Africa.
Got it. Okay. And our last question, was there any data about BMI/obesity or use of exogenous herbal testosterone levels in South Africa?
So for this current study, or for this cohort, there was no data for that. However, for the study that is still ongoing, they are collecting that data. So we will have more information in the next publications.
Thank you so much for this great presentation. The rest of the comments in the chat are just saying how insightful and very interesting your presentation was. So,
Thank you.
we will now have Dr. Sung present. Hi, Dr. Sung.
Hi, can you see my slides?
Yes, so there we go. Thank you.
Awesome. All right. Well, thank you everyone for coming to this webinar today. We're now going to switch gears to genetics. So as Vanessa very kindly introduced, I am Dr. Pamela So from her lab at the University of Sydney. And today I'll be telling you about some of the work that I've done on inherited germline risk, specifically the need for an African-specific polygenic risk determinant for prostate cancer. So what is a polygenic risk score? By the name, poly means multiple and genic referring to genes. So it is a score given to individuals in a population based on how many risk alleles across the genome they carry for a disease or trait. And really the goal is to stratify those individuals at high risk of the disease or aggressive disease so that we can detect these individuals earlier or give them treatment earlier. And how is a polygenic risk score calculated? So first you would get data from other genome-wide association studies. They would have a list of variants with an associated risk allele, and they will have effect sizes. So this can be positive, meaning that it increases risk of the disease, or it can be negative, meaning it could be protective against the disease. And then you would look at those variants in your own data. So for example, here I've got 2 samples, and sample 1 has 2 risk alleles for variant 1. It's got none for variant 2, and it's got 1 for variant 3. And this is the formula. So it's the sum of the number of risk alleles times the effect size divided by the ploidy, which in this case is 2, times the number of variants. So then you would score each sample this way. And then in this scenario, sample 2 has a higher risk score than sample 1. So they're at increased risk of the disease. Now, polygenic risk scores, or PRS, has been really successful in European populations for prostate cancer. This is an example from McHugh et al., which was recently published in the New England Journal of Medicine. And they show that among 186 patients, only 30 samples would have gone into biopsy based on the cutoff of PSA levels above 3 micrograms per liter and PIRADS above or equal to 3. Meaning that 156 patients would have had a misdiagnosis if not for the polygenic risk score. So this is really promising. And why is this so successful? This is because of really strong statistical support. So this PRS was constructed based on 130 variants associated with prostate cancer in a study with over 140,000 European men. It's a massive study. And unfortunately, we don't have that luxury in Africa. So when I published my study in 2023, this is what we call the Genome-Wide Association Study Diversity Monitor. It just shows you the diversity of all GWAS studies across the globe and tells you the diversity. And we saw in 2023 that Africans only contributed 0.16%, African Americans and Afro-Caribbeans was 0.49%. And you see that Europeans contribute the large majority at 95%. In 2025, promisingly, African American and Afro-Caribbean samples have increased in proportion. However, African studies are still stuck at 0.16%. And as Winstar has mentioned in his talk, prostate cancer studies are quite sparse in Africa. And that's also true with genetics. So when I published my study, there were only 4 prostate cancer GWAS studies that had been done in Africa. And to this date, I think there's only 1 extra study, and it has used samples that I've labeled here as well. And you can see from the numbers as well, we have really tiny numbers compared to the European studies. We definitely don't have 140,000 European men. In red here, I've just got NOMAD data labeled. So NOMAD is an online resource of publicly available genomes, which are meant to represent the global diversity. However, you see that Southern Africa has a very limited representation. So in my study, I was assessing the PRS based on these 2 studies. So the first one was a multi-ancestry genome-wide association study that identified 269 risk variants. And the second study was focused on African ancestry, which identified 9 new risk variants. So that first study, so they did GWASs for European ancestry, African ancestry, and a multi-ethnic ancestry, which included Asians as well. You can see in the European ancestry, they have pretty big numbers, 85,000 cases, 91,000 controls, and we get many peaks across the genome. So all of these points are a significant association. And then in African ancestry, we see we only have about 1/10 of the cases and controls. And it seems like it's much lower signals compared to the Europeans, and even the axis, look, this is half of what the Europeans have. And what they did was they generated a 269 risk SNP set that represented all these associations using the multi-ethnic ancestry GWAS. And you can see that this multi-ethnic GWAS largely reflects that of the European ancestry signals. And then in the latest study that looked at African ancestry, what they did was they did a conditional GWAS based on those 269 risk variants. So what they did was they masked those signals to see what else would appear significant. And they found 9 novel risk variants. So they added that to the PRS, and it was able to stratify aggressive versus non-aggressive prostate cancer in their African ancestry cohort. And if you look at the distribution of their population, 16% of their cases are African and 4% of the controls are African as well, whereas the rest of the samples are all African American. And now generalizing all these samples as African can be a little bit of an issue. And why is it an issue? So this is what we call an admixture plot. This is what companies such as Ancestry.com and 23andMe would use to determine what proportion ancestry you are. And how the program works is essentially you give the program your population and you tell it, okay, I want you to group the population into 2 groups. So if I ask for 2 groups here at k equals 2, each column is an individual. So if I ask it for k equals 2, we see Chinese and European people split off from Africa, in purple. If I ask for 3 groups, then we see Southern Africa pops up, 4 groups, West Africa, 5 groups, Central Africa, 6 groups, East Africa, and 7 groups, West Africa splits again. And you'll notice that Chinese versus European populations still do not split at k equals 7, meaning that the diversity between Chinese and European is much less compared to region between Africa. And you'll also notice that African American and African Caribbean samples primarily have Western African ancestry and a little bit of European admixture, which makes sense. They, however, do not represent the rest of Africa. They have very little Central, Eastern, and Southern African contribution, as you can see from the plot here. So really, genetically, Africans should not be generalized into one population. There's over 2,000 ethno-linguistic groups across Africa with different languages, cultures, traditions, and lifestyles. So we can't group them all into one. So in my study, what I did was I tested the PRS in a cohort of Southern Africans, and we were unable to find significant association between the PRS and aggressive disease in our cohort, meaning that I cannot distinguish aggressive from non-aggressive disease using just these 278 variants. And this is in line with other studies as well. Southern Africans have the poorest prediction accuracy, which was found in this study that looked at the previous version of the PRS with 269 risk variants. They found that it performed the best for Europeans, moderately for West Africans, and worse for Southern Africans. So if those 278 sites don't explain it, then where should we look? So this was a exome-wide association study that I did in Black South African men. What we did was a case control analysis first, and we found hits in genes such as HCP5, which is involved in metastasis, and RFX6, which is a transcription factor. And this one in particular has been associated with decreased prostate cancer risk in men from Ghana, Japan, and China. And next, I also did a aggressive versus non-aggressive prostate cancer association analysis, and we found genes, variants in genes such as MKI67 as a marker of proliferation, and PCSK6, which is correlated with ERG tumor status and ISUP grade. So these gives us some hints of where to look next. We don't have as strong signals as you saw with the other studies. We definitely don't get those clean peaks. But it gives us some idea of where to look next. We definitely need to do more in a bigger cohort. So some caveats, the risk variant set has actually now expanded to 451 variants as of 2024. In that study, I believe that they added a lot more African American data as well. So it'll be interesting to assess how that has changed. So second of all, as I said, we have a small sample size, but also the exome array design is European skewed because the arrays are designed based on known information, and there is a lot unknown within Africa. And lastly, germline prostate cancer risk could also differ across Africa. So this is one example. So it's a HOXB13 risk variant. that's present in Western Africa, but it's absent in Southern Africa. And the hypothesis is that this could have arisen after the Bantu migration between the border of Nigeria and Cameroon, spreading eastwards and southwards about 1500 to 4600 years ago. So just to conclude, current polygenic risk scores for prostate cancer do not perform well for Southern African men. We identified regions in the genome associated with prostate cancer risk and aggressive prostate cancer. And lastly, more studies are needed in a larger cohort with data that is more inclusive of Southern Africa's diversity.
The presentation, Dr. So. I really learned a lot. I really liked your graph about the genetics and the pictures at the bottom. That really
Thank you.
the fact that that Africans are not all monolithic. So.
Yeah, it's so diverse.
Exactly.
There's a lot we don't know.
Yeah, so it just opens up the realms for more research and understanding.
Yeah, for sure.
All right, so I'm checking the chat. So if anyone has any questions, comments, and or feedback, if you could write them within the chat for questions for Dr. So. We'll give the audience 30 seconds. If not, we'll move on to our next presenter. So some comments coming in, letting you know what a great job and presentation you've done.
Thank you. I think we'll move on to Vanessa. I'm worried about time running out.
Yes. All right. Thank you.
I can take questions at the end if needed.
All right. We'll move on.
Thank you.
Please. Thank you.
Hi.
Hi, Dr. Hayes. All right.
Hi.
I'll be presenting your slides.
If you can. Yes, please. Right. So I'm actually going to follow on from what Pam just told you, and I'm going to talk about another aspect of risk profiling. Next slide. So what Pam just spoke to you about was looking at common variants that are found across the human genome. So these are variants found in many people, but their effect sizes, their effect on risk is not as high. But as Pam showed you, you can have a polygenic risk score where you mix a lot of variants together to create this risk profile. But there is another kind of variant that we find associated with disease, and these we call the rare variants. Now, these variants are not common in the population. They're very low, actually a percentage, usually less than 1% of the population. But when you have one of these variants, their risk for disease is very, very high. So therefore, that's the reason why you wouldn't have so many people having them. And we're very interested in these variants because usually they occur in men who diagnosed with very aggressive disease at a younger age. They probably have a family history of prostate cancer, or they can have a family history of breast and ovarian cancer in their family. So in this kind of work, we want to look for these, as opposed to what Pam was doing, look for these rare variants. And of course, in this kind of instance, you're not going to look at thousands of people. You're actually going to tailor your research into men with a particular phenotype that could be carrying these rare variants. Next slide. So if we take these two gentlemen, say, for example, with high risk and very, very high risk in red or high risk, we would take their blood and we're going to look for these variants. So where are they likely going to be? They are most likely going to be located within genes that are functional within the human genome. But only less than 2% actually of the human genome actually codes for genes, and we have roughly 20,000 genes within the human genome. So 98% of it doesn't code for genes. Of those are roughly 20,000 genes, roughly 200 of them are what we call DNA damaging or DNA repair types of genes, which are the ones most commonly found to be pathogenic in many cancers, including prostate cancer. So from these 200 genes, the community has created what they call a gene panel, and that's roughly 20 genes that they say is commonly mutated in roughly 11 to 20% of people with very aggressive prostate cancer or family risk of prostate cancer at a younger age. And we have these panel. So what's, next slide, please. So what Kazem Gebi, Dr. Kazem Gebi in our group did was he looked at these 20 genes in our Southern African cohorts compared to an Australian European cohorts that were matched for clinical presentation and aggressive clinical presentation and age as well. And what he saw was that while these 20 genes, variants within this 20 genes could be had a prevalence rate of 17% in our European population, it was only roughly 5% in our Southern African population. So next slide. So the study basically raised three important questions. And raised three important questions, three problems that are being addressed, that have been addressed by members of our team. So the first question was computational tools. Are we using the right tools? And this was addressed by a PhD student, Kangping Zhou. The second question is, have we got the right candidate genes? And this Dr. Kazem Gebi has continued to look at with his paper just accepted in Nature Communications. And the third question we have is, are we looking at all the variants we should be looking at? Are we missing some very large, impactful variants? And this study has been addressed by Ting-Ting Gong, Dr. Ting-Ting Gong, and her paper was published also this year in Nature Communications. Next slide. So I'm quickly going to go through each of these studies. So in the first study that Kangping did, looking at computational tools. So he matched two groups. In orange, we have Africans. In blue, we have Europeans. So these were men matched as best as possible for their pathologies, their presentation, numbers as cases. Now, as Pamela showed you, African men have greater genetic diversity. So this also equates into the number of variants that you will actually see in the human genome. And in this case, the genes also cross the human genome. So African men in Southern Africa will have a 1.6-fold greater number of variants. And even when you look at rare variants, so that's the variants under 1% in the population, they still have a 1.2-fold greater number of these rare variants. So then what Kangping did was he went to see how many of these rare variants are potentially pathogenic. So we call them pathogenic. They're likely to have a clinical impact for the patient. And then you will notice that it flips. Even though African men had more variants to look at, it's the European men that had more pathogenic variants. And the reason for this is because the data in the databases that define clinical variants, as well as the criteria that have been established based on European data. So it's purely that there's data lacking for African men. So what we end up is a group of variants we call variants of unknown significance. And while 48% of the variants found in our Europeans were of unknown significance, almost 78% of the variants Kangping found in the African patients were of unknown significance. So this is really important. That means the clinicians, well, we find variants, but we don't know what to do about it. We can't provide any clinical impact for the patients. Next slide, please. So taking these variants, they are now of unknown significance. The community has established a number of tools. In fact, there's 54 computational tools out there that are used to try and predict if these variants of unknown significance are potentially pathogenic. Now, Camping has ranked them from left to right, all 54, and he's ranked them from how well they've done within in orange again, the African cohort versus the European cohort. And you will see there's differences between the tools. Now, in the middle, I've boxed the two most common tools. And you will see that even though these are the most common tools used and they perform fairly well in European populations, they don't perform as well in our African populations. And in fact, on the very right, there's a new tool that came out that has been sort of given the golden star as the best tool, but you can see it actually did not perform. It performed one of the worst in our African populations. Next slide. So in the end, what Camping has done is he's created a 10-tool workflow where he said, what are the best tools that predict pathogenicity for Africans and what are the best that define them in European patients. And while there are 6 tools that can be shared across the ancestries, there are 4 each that are very specific to each ethnicity. Next slide. So now moving on to candidate genes. So this is a study that Dr. Gaby's been doing. So here he looked at 217 African patients. Majority of them are Black South Africans, and we also had a cohort of majority African American ancestral Africans. So as Pam mentioned, having more West African ancestry. So again, he looked at the common panel of genes and found that it only predicts prevalence for about 66% of our cohort. He then looked at which of those genes were highest ranked for carrying pathogenic variants. And actually the top ranked gene is the same one for this gene panel that's actually found in Europeans, which is BRCA2. But it was very interesting that 6 of the genes in the panel were completely silent in our African populations. And this is important because remember I said at the beginning that Africans are more likely to carry variants, including rare variants, than people of European ancestry. Next slide. So then what Kazim did is he took all 200 of these genes and he looked at all of them, so independent of whether they're on a panel or not. And he identified 78 DNA damage repair or mismatch repair candidate genes. He then developed a model based on variant features, clinical features, and features of the tumor that I don't have time to go into. And he could rank these variants, and you can see the genes at the bottom ranked, and all the ones in blue or dark blue are new to the study. The ones in green are the ones that are currently on the panel. And you can see there's this vast array of new genes that we really need to consider to create an African relevant panel for germline testing. Next slide. And then we wanted to address the last question. And this is, are there large impacting variants that are yet to be found? Now, all germline testing or genetic testing currently performed ignore what we call large structural variants. So all of this work is currently looking at what we call single nucleotide variants or small insertions deletions. So that is just single letter changes within the DNA code, or a few letters up to 50 letters maximum. But we know in the human genome there can be these very large events, large deletions, insertions, duplications. And we know, although they're not as common as these small events, when they occur, they have a large impact on your DNA. And we also know that you can inherit these events from your parents. So no one actually had been looking at these as potential to identify as potential pathogenic events in prostate cancer. So this is what Ting Ting went ahead and did. Next slide. So this is how she does it. One of the reasons why people don't look at these events is they are very difficult to detect because using the technology that we mainly use, which is called next generation sequencing or short read sequencing, these events are larger than the length of the DNA that we actually get off the machine. So Ting Ting has to use computational methods to infer if this event is actually there. And you see two examples at the top. Those are indicating what reads, sequencing reads. And from that on the left, she can see this particular, these reads are showing a deletion event that has actually deleted the middle part of this gene, and she's schematically drawn it at the bottom in blue. So a big portion of this gene that is incredibly important in maintaining cellular growth has been deleted. On the right is another example where part of the gene has actually moved from one chromosome to another and inserted itself in to another chromosome. That's called a translocation, again disrupting a very critical gene. So let's go and see what Ting Ting found. Next slide. So using this approach, Ting Ting identified only 3 variants that are known to the databases, the ClinVar databases. So they're known to have a clinical impact. But in total, she found 15 candidate genes and another 4 potentially extra ones that we're not quite sure on. And I've pointed out one there where I want to link back to Kazim's data when he was looking at the small variants. You'll remember that I said there were 6 genes that are on the gene panels that were highly, highly conserved in our Africans. We didn't see any variation there. But when Ting Ting did her approach to look at these large events, she did find a deleterious variant in the gene called MLH1, which is a DNA damage repair gene known to be very important in prostate cancer. But this would have been missed if we hadn't taken this approach. Next slide. So just looking closer at this example, she found it in one African patient. It was a big inversion, so inversion event that disrupted MLH1. So you can see large part of the gene has gone missing. This gene was definitely inherited in this patient. We can tell that by the allele frequency. The patient was 64 years of age, which is very young for our African cohort. And the man presented with aggressive disease, so a Gleason grade, a pathology ISAP grade 4 and a PSA over 20. So a high-risk disease. We could also show that this actual inversion event caused this gene not to be able to be expressed. So what you see in the plot on the right is decreased expression of this gene in this patient. Going on to the, oh, don't know what's happening there. Next slide.
My apologies.
Sorry. Next slide. And I've just got one more example before I end up. This is another example I just want to show you. So not all genes that Ting Ting found are actually known DNA damaging repair, but there could be other types of genes. In this case, she found a common tumor suppressor gene that's usually involved in the tumor as it evolves in prostate cancer. But this time she found an inherited event. Next slide. And in this case, she actually found this, it was again an inversion event, a very large, that disrupted the FOXP1 tumor suppressor gene. So this time she found it in 2 African patients. It was inversion that led to loss of function of this gene. Again, we could confirm that it was inherited. Interestingly, one of the patients was 57 years of age with the highest risk pathology of prostate cancer, an ISAP5, which is very unusual. We've seen our cohort average age was 70. The other gentleman also had aggressive disease. And you can see the very high PSA levels there, 75 and 591. And again, we could show that this disruption did lead to loss of function because this gene is no longer expressed. So in conclusion, our concluding slide, from this work, we can show that we need to establish African relevant genetics testing criteria, which needs to address computational workflows to predict pathogenicity that are African relevant. We need African relevant gene candidates for panel optimization. And we need to consider these very large, what we call kilo to megabase long inherited events for their potential role as well within genetic testing. And I just want to end with the last slide, which is the heroic team. This was our meeting in Kenya this year. And you heard two talks today from the urology and pathology team at the top of the left, from Winston Tabojo. You heard two talks from the genomics and data science team on the right top, myself and Pamela. And in the future, you will hear from our other two teams, our public health and awareness team on the left, and our exposomics and endocrinology team. Thank you very much.
Great presentation, Dr. Hayes. I love the pictures and the diagrams. So thank you so much for your presentation.
I know we're over time, so people may be tired.
No, you're okay. We are actually receiving some questions within the chat. So, so our first question that we have for you, Dr. Hayes, is first question is, they may have missed this, but they wanted to ask, what is the percentage of your African participants who are in Africa and Africans in diaspora?
Okay. Yeah, so so for the majority of the three studies I just presented, they were mostly Southern Africans, Black South Africans. And as Pam showed you, quite different genetic ancestry to the rest of Sub-Saharan Africa and African American populations. But in the study where we looked at the small variants across the human genome, we also looked at a small number of African Americans. The reason we've only looked at a small number up to now is that our analysis needs what we call whole genome data. And there isn't a lot, even for African American data, there's not a lot of comprehensive whole genome data available yet. So that's why we're working very closely with our team in Chicago, who are also collecting information. So we will be able to tell you more in webinars to come about how this differentiates across the diaspora.
Thank you. Follow-up question is, if any, what are the differences that you're seeing? And then thank you. So, so I guess that kind of, your answer prior helped cover that follow-up question. Semi-similar question, but given the diversity across Africa, as you mentioned, should we be more micro in our approach and focus on regions in Africa rather than continent-wide gene candidates?
Absolutely. If you actually look at the Globicon studies that Winstar showed you right in the beginning, the very first slide, if everyone can remember that picture, even if you look at Globicon's records of mortality rate and incidence rates, it differs across Sub-Saharan Africa. So we really have to consider Southern Africa as unique from Central Africa and unique from East Africa and West Africa, because we don't only see genetic differences, there's environmental differences, cultural differences, and as Pam showed you, that that rich diversity needs to be considered.
Next question we have is there work similar to work in breast cancer that is looking at the tumor microenvironment and VUS in prostate cancer?
So in what we were presenting today was the germline stuff. We haven't presented here today anything to do with the tumor itself. That will come in later webinars. So we're just focusing on the inherited. And there actually is some work in the breast cancer that has been done, but like the prostate cancer, it is very, very sparse. But I can also tell you in Southern Africa for breast cancer, again, we see the predominance of BRCA2, and again, BRCA1 doesn't seem to be very active, which is a gene in Europeans that is highly mutated and quite a strong candidate on the germline testing panels. So we definitely I don't work on breast cancer, but just knowing the field, that's starting. But unfortunately for cancer research across the continent, we've got much to do. There's a lot of work still to do.
Yes, I bet. Most definitely. All right, the final question that I see in the chat. So again, a lot of comments commending you for the great presentation. So our last question that I see here is the MLH1 and FOXP1 variants that were found, were they from Black participants in the US or South African?
Those were all South African. Yeah.
Great. So just wanted to ask any final rounds of questions. I know there was a question for Dr. So that was asked after her presentation, so I'm not sure if she's on. We can add her on stage too. And Dr. So, the question that was asked for you was, are there any differences between African men and other populations?
Yes, there would be lots of differences, from culture to tradition to genetics. As you saw from my talk, there's very different population and population. So there are many differences. Yes.
Right. Okay. So I just wanted to make sure we address this question as well.
That's okay.
Adding just if it's okay with all the panelists to add them onto the stage and to all go in. It's okay to add that. Just wanted to say, ask the audience if there's any final rounds of questions for our presenters. So if so, feel free to ask them in the next 30 minutes. Okay. So another question is, and it might be for Dr. Hayes, but in your experience, how can we increase NGS in Africa?
So the way I think we need to do that is, and the way we've been tackling this question in our team is through collaboration. The advantages now is that the world has become much smaller by the fact that we can connect this way, the way we're connecting tonight in Australia, in the morning in US. So for example, Winster, Tobolgo, they're 100% part of our team. They connect to our high performance computer infrastructure in Australia. And we are able to communicate on a weekly basis as if we are one team, but we are sitting in different areas. So I think technology has really brought Africa to the world. And so therefore, it makes NGS absolutely possible. And we just have to embrace it. The rest of the world has to embrace why it is important. Why is it important for all of us? Because we all come from Africa. So that's where our roots lie. And that's why there's such rich diversity within Africa, because for Pam and I, whose ancestors left Africa, that's why we can't even separate on Pam's graph. We look exactly the same to the computer because we lost all that rich genetic diversity. So actually outside of Africa are more rare genetic diseases because of that loss of diversity. But on the flip side, diseases like prostate cancer, who are diseases of older age, have predominated and not been selected out within Africa because of the age distribution of this disease. So we're really going to understand the disease better for everyone if we start including everyone in the world. And I think bringing in NGS to Africa is absolutely feasible, and it is increasing.
Great response. Just wanted to thank One more final question, I guess. Sorry, Dr. Hayes. I know we kind of went over the 1 hour, but we just have a lot of audience members curious
Technical.
to hear you all present. So, what future, and this is for any, mainly probably aimed for our geneticists, but what future of genetic risk cancer in Africa? What future of genetic risk cancer in Africa? Your points of view.
We need a lot more data.
Yeah.
Especially for Pam's work. I mean, that's a numbers game. So data. We need to get more people on board. We need to get the funders on board. One of the biggest problems with Africa being left behind within the whole genomic era has been no money. It's still expensive to do what we do. It's obviously a lot cheaper than what it do. The first genome we sequenced cost us $2 million for one person. And that was in 2008 to 2010. It took us 2 years just to look at one genome. So we've come a long way, but still, it's big money. It's a lot of computational power. You do need high performance compute infrastructure. So that's why we need to work together.
I will also add that a lot of the tools that we use are based on European data sets, and they don't perform very well for African data. So that's also something that we need to consider going forward.
Thank you all, Dr. Hayes, Dr. So, Dr. Mbuki, Tibogo. Thank you for all this, your presentations, for being open to answering questions, for staying a little bit longer within this webinar. This great collaboration within each other just allows for us to learn more about your research, as well as a better collaboration between USA and Australia teams. So just all that global work that we want to help collaborate together. So Dr. Denham was saying that the two heroic groups may also need to look at standardization and uniform measures / data collection for integration that we should discuss. So thank you again, Dr. Hayes, Dr. So, Dr. Mbuki, Tibogo. We will end this webinar with our theme song. So thank you to our audience, especially for joining in, for listening in, for all your questions, comments, and feedback. We really appreciate it. All right, thank you, everyone. And we'll play our theme song. All right, thank you, everyone. Have a great one. Bye.
Bye. Thank you.
Bye.